Alright, ladies and gentlemen, welcome back. We're going to continue our discussion on enzymes, and today we're going to talk about factors that affect how well an enzyme is able to catalyze a chemical reaction. So, remember that enzymes are specialized proteins, right? And so, in order for a protein to do its job, it's very dependent on the way that that protein folds and the shape that it creates once those folds happen. And so, the folding of that protein, or in this case more specifically an enzyme, can really get affected by certain things in its environment. So we're going to talk through three different classes of factors that can affect how well an enzyme is actually able to catalyze or speed up that kind of a chemical reaction. And let's start with the environmental conditions themselves. So, in order for an enzyme to do its job, it has to be folded appropriately, just like any other protein in your body, which means that in order for these enzymes to work and do their job at their optimum, your body has to maintain homeostasis. We have to keep those stable internal conditions, whether we're talking temperature or pH or salt concentration. Because if we don't, the further and further away from homeostasis your body or that cell in that organism becomes, the more likely we are to get a protein, or an enzyme in this case, to denature, completely unravel, lose its functional shape, and once that happens, there's no active site for a substrate to bind to, which means there's no way for that enzyme to continue catalyzing or speeding up that chemical reaction. And in a lot of cases, those chemical reactions then just don't happen at all because they are so slow to start naturally. So if we take a look, alright, a lot of times we'll look at reaction graphs for enzymes and there'll be these big peaks. And what I'm looking at is at the peak of each of these diagrams is the optimal location. This is where, if I'm looking at temperature, this is the best temperature that I'm going to get that enzyme to work at. It's going to be doing its maximum job. It's going to be doing the best job that it's possible of doing. Alright, and that's what it means by that rate of reaction over here. Same thing down here. Alright, peak of the graph, but now I'm looking at pH instead. So one of the things I wanted to point out while we were on this particular slide is that this is the optimum. This is where the enzyme is working its best. It's catalyzing the most chemical reactions it can in a particular period of time. It's breaking down the most substrate it can at that particular period of time. But if I get away from that optimum temperature or away from that optimum pH, notice that the reaction is still happening. It's just happening less. Alright, so let's assume that at that optimum for this particular human enzyme, so we're talking about body temperature, it's about 38 degrees Celsius. That enzyme is able to break down 100. So if I'm at 100 molecules of sucrase here at its optimum, as I get lower and lower and lower in temperature, maybe here I can only do 100 molecules of sucrose that I'm breaking down. I get further and further down here, maybe it's 50, right? And maybe I see something similar on this side, but it happens to drop off a little faster in temperature, so 100 here, 50 here. So that chemical reaction is still getting catalyzed. It's just not happening as efficiently as it could if I were at that optimum condition. So as homeostasis begins to get lost and getting further and further away from that optimum body temperature, the enzyme's still working, but it's slowly losing its shape. Alright, and so the less perfected shape I have of that enzyme, the less effectively it works. It doesn't mean it completely shuts down until I get way off the graph. So within this black line, that enzyme is still working. I'm still breaking things down, but once I'm, say, maybe up here at 60, that enzyme is completely unraveled. There is no way it's doing its job anymore, and so I'm not catalyzing anything. That's the big difference. So if your enzyme changes shape, then the reaction that the enzyme catalyzes stops. So if I completely lose homeostasis and I get way out of that set point, way away from that optimum temperature or optimum pH, or whatever the condition may be, that substrate no longer has an active site that is able to fit the substrate that it works, and so the chemical reaction stops happening. So what does all this mean? Well, if you've got a situation where you're missing an enzyme or an enzyme isn't functioning properly because that homeostasis is lost, it really becomes a big problem, because when we look at chemical reactions that are going on in your body, it's very rare for it to be one product that gets made from one set of reactants and the whole thing's done. Instead, most of your metabolism is these big series of chemical reactions pathways, and in fact most of them are way longer than this. But this is just an example. So if I have a situation where in order for my body to make that product C that I really ultimately need for my cells to work, but my body has to take in A, so I've got to go from A to B. One enzyme's going to catalyze that reaction, but then I need to go from B to C, and I've got the second one. If I've got a situation where enzyme 2 doesn't work, this can be a really big deal. So I've got a couple of situations that might potentially get set up here. Well, first off, I'm not getting product C regardless, right? So I may be able to get from A to B because this enzyme still works, but this enzyme, either I'm born without it, I've got some mutation that makes the wrong shape, or something has caused it to get destroyed. Well, now I'm not making that essential product that I actually need, something like maybe insulin, for example. All right, and insulin job is to control your blood sugar based on the amount of carbohydrates that you take in. So if your body's not able to deal with properly regulating that glucose level, that could be dangerous to your body. People with diabetes, that's their problem. All right, they're not able to properly utilize that insulin or make it in the first place. Another potential option here is that while I'm not getting that end product that I ultimately need, which is also a problem, I may also get to a point where if I can still do A to B, but B is toxic to my cells, and now I can't break down that toxic product to get what I really needed, this accumulates in my cells, and that can also be a problem. Something called PKU disease is an example of that sort of situation. In fact, it's a disease that we typically test newborns for. Why? Because it's actually something that we can control with diet, so long as they stay away from certain amino acids that their bodies aren't able to break down, they don't have any problems. Okay, but they happen to ingest it, they're missing this enzyme, and so this toxic product builds up and it can make people very sick. Alright, so another potential factor that can influence how an enzyme works has to do with these molecules called cofactors and coenzymes. And these guys are compounds that exist in your body. They're not proteins, but they'll actually bind to the enzyme, a lot of times binding to that active site and helping to bind a substrate even tighter. So they're helper molecules, and they help to assist in that chemical reaction, make the enzyme work and fit its substrate just a little bit better. And a lot of these tend to be your vitamins and minerals, so things like zinc and iron. For example, iron, has to be present in hemoglobin. It's a cofactor. And that hemoglobin, in order for it to pick up the oxygen in your red blood cells so it can carry the oxygen to the cells of your body and drop it off, if you don't have that iron, the hemoglobin will still attach the oxygen, but it will not do it as efficiently as it could with the iron present. So if you ever hear somebody who talks about being anemic, Alright, it's a medical condition where their body isn't able to take iron up out of their diet well enough, which means they're lacking that iron attached to that hemoglobin, and so although their body can function and they'll pick up oxygen and they'll drop it off at their cells, it's not working as well as it could. And so as a result, a lot of times they'll get winded faster, they'll be tired faster, they may bruise easier, and a lot of that's simply because they're missing that extra to help those enzymes work just a little bit better. The last factor that we're going to discuss, at least for this year, has to do with enzyme inhibitors. So one of the things about enzymes is that we don't necessarily need them to be functioning 24/7. If I have a lactase enzyme in my stomach, but I haven't had any lactose, meaning I haven't had any dairy that day, then why do I want to have this lactase enzyme constantly looking, constantly searching, constantly trying to function if there's nothing for it to break down? It's just going to wear out that enzyme faster. So we can regulate your enzymes using inhibitors. Alright, and that's one important way to understand how these work. Also, inhibitors end up getting involved when we talk about toxic substances. They'll sort of mimic that substrate and get in the way of that binding of that substrate, which is what ultimately makes you sick. Flip side to that, we can also use it, medically speaking, to make drugs and medications to block things like pain receptors. Alright, and if I can mimic that substrate, prevent the enzyme from doing its job, I might be able to make you feel better as well. So you've got two different types of inhibitors that work. They're either competitive or non-competitive, and it really comes down to where they bind on your enzyme itself. So if I'm talking a competitive inhibitor, here's my enzyme, right? That competitive inhibitor actually sits right in your active site. So here's your active site here. That competitive inhibitor literally competes for the exact same spaces that substrate would want to be in. And so it blocks its entrance. If it can't bind, that substrate never gets broken down. You never get the products as a result. So they're literally competing for the same active site. A non-competitive inhibitor, however, enzyme non-competitive inhibitor binds to a secondary site on your enzyme, and as a result, that active site changes shape. So the binding of this guy causes the entire protein shape to change, and once again, that substrate comes in, shape's not there to fit into, and so it can't actually fit, and nothing happens there as well.